My name is Komlan Atitey. I'm employee of NIH working in biostatistics and computational biology branch. Today, I will talk about multicellular and multiscale modeling of proliferative immune response under joint synergistic and antagonistic cytokine signaling. The presentation will cover 5 points. Firstly, multiscale multicellular quantitative evaluator. Secondly, T-cell activation protocol. And third, mathematical model, follow with the result, and finally, the conclusion. I'm working in the research group of Benedict Acham, including Raul Mendez. Since there are prospects for use of interleukin-2 and interleukin-4 in the treatment of autoimmune disease or infection, the contribution of T-cell cytokine production to the pathogenesis of autoimmune disease is still a matter of debate. The major key players and biological process in the proposed MMQE for overall immune system response are modeling T-cell activation with a dendritic cell, modeling the variation of antigen, modeling the dynamic of B cell, modeling the proliferation of plasma B cell, and the dynamic of T cell, and finally, modeling the interaction between IL-2 and IL-4. The first activation protocol refers to the dendritic cell playing the role of antigen-presenting cell. The second activation protocol refers to B cell activation. The third activation protocol is managed by the joint activations of dendritic cell and B cell. We firstly model the interaction between interleukin-2 and interleukin-4 for immune response. We illustrate with ordinary differential equations the complex interactions between the dynamic of cytokine IL-2 and IL-4. On the first hand, the proliferation response to IL-2 stimulation is mainly controlled by three factors. The first is the initial concentration of IL-2. The second is the density of receptor of interleukin-2 on the surface of the T cell. And finally, the durations of the cell exposure to interleukin-2. On the other hand, IL-4 binding to its receptor have inhibitory effect on interleukin-2 induction. Consequently, the mathematical modeling of the dynamic of IL-2 and IL-4 in our work is based on all the known interactions between IL-2 and IL-4 to predict with a high accuracy the dynamic of T cell and B cell immune response. We define the dynamic of the pathogen distribution in the immune system as a random process, which is modeled as a log normalized density function. Since there are possibilities that the helper T cell get activated independently by DC and the B cell or both simultaneously, we assign a variable to the varying nature of activation protocol of T cells. And also, we model the dynamic of B cell population's immune response as a function of a B cell activation, disappearance, or differentiation into more antibody. And finally, the plasma B cell is modeled as an output result of B cell differentiation. We first evaluate our MMQE model in terms of the interaction between IL-2 and IL-4. IL-2 show a rapid association rate with its receptor, and IL-4 present a slower association rate with its receptor IL-4R. The proliferation of T cell and B cell increase to a maximal level, followed by degradation, regardless of the immune system activation agent. However, the maximum number of T and B cell produced during their proliferation life cycle depend on the activation agent. For example, B cell activating agent lead to a high maximal level of T and B cell proliferation. Also, the proliferation level of plasma B cell reaches steady state early when the activation mechanism is regulated by B cell agent. Also, the proliferation level of T cell, B cell, and plasma B cell entirely depend on the interaction between the cytokines IL-2 and IL-4. So, T cell, B cell, and plasma B cells together display a high level of the immune response, which is associated with low concentrations of interleukin 4 versus high concentration of interleukin 2. T cell, B cell, and plasma B cell present a higher level of proliferations when the immune system is simultaneously activated by dendritic cell and B cell. This figure summarizes a direct head-to-head comparison of proliferation of a CD4 T cell between CD40L activated B cell and mature DC with regard to presenting antigen to the naive T cell for activations. It showed that a T cell presented a high proliferation with B cell activation than a DC activation. We also evaluated experimentally the proliferation of T cell under 3 different conditions. We observe a higher T cells productions in the absence of interleukin-4 stimulations. It verifies how IL-4 downregulates interleukin-2 for T cell proliferation. 2 additional mouse experiences were made under 2 different conditions. The results show once more again the downregulation effect of interleukin-4 or interleukin-2 in T cell activation and proliferation. The simulation result and validation result lead to conclude that MMQE is well suited to model the adaptive immune response in terms of the regulations of multiple cytokines in relation with different activation protocol of the immune system condition on dendritic cell and B cell. This work is funded by NIH Division of Intramural Research. Thank you.